The Shroud of Turin is this amazing relic. It shows a very faint sort of image of a crucified man, and the crucified man calls us to remember Jesus' death and resurrection.
What if a centuries-old religious relic was quietly preserving a biological record that tells a story far beyond faith or myth? Imagine uncovering strands of ancient DNA from people across continents, all trapped within a cloth that might have touched the very places and people it claims to represent. The Shroud of Turin, long debated as either sacred evidence or medieval forgery, hides secrets no one saw until modern genetic tools revealed an astonishing trail spanning the Middle East, Europe, Africa, and Asia. The DNA mystery of the Shroud. The Shroud of Turin has long captivated believers and skeptics alike, but recent scientific investigations uncovered something no one expected: ancient DNA fragments hidden deep within the linen's fibers. At first glance, these genetic traces seemed like a natural result of centuries of human contact. Pilgrims, priests, and custodians all touching, carrying, and preserving the cloth throughout history. But as researchers mapped the DNA markers, a surprising pattern emerged. The genetic material didn't just reflect one region or group. It spanned continents, connecting the fabric to populations from Europe, the Middle East, North and East Africa, and even South and East Asia. This astonishing diversity of DNA raised immediate questions. How could a single piece of linen carry genetic traces from such a broad geographic range? If the shroud was indeed a medieval creation from Europe, the DNA should primarily reflect that area. Instead, the data suggested a far more complex history, one of prolonged contact with many peoples along ancient trade routes. Some researchers started to consider that the cloth might preserve a silent biological archive, a timeline written not in ink, but in cellular fragments accumulated through thousands of years and countless encounters. Scientists focused on mitochondrial DNA, a form that survives longer than typical genetic material and can indicate geographic ancestry. Using highly sensitive micro-vacuum tools, they carefully collected the microscopic particles trapped within the linen's threads, preserving dust, pollen, and tiny biological fragments. Over weeks, powerful computers processed millions of genetic sequences, comparing them against global DNA databases with meticulous precision. The results didn't point to a single source or moment in time. They unveiled a sprawling narrative layered through millennia. The implications of these findings unsettled long-held assumptions. If the shroud were a medieval forgery, gathering such wide-ranging genetic material would require an impossible feat of collection and preservation. Instead, the more plausible explanation is that the cloth traveled extensively, touching diverse peoples and places over centuries. Each contact left behind microscopic biological traces, building a complex genetic mosaic within the fabric. This revelation shifts the shroud from a static relic to a living document chronicling a vast human journey. Yet the mystery deepens. The DNA alone does not explain how or why so many genetic fragments came to coexist in one place. Could this accumulation simply result from the relic's movement along famous pilgrimage and trade routes such as the Silk Road? Or does it hint at a story spanning the entire ancient world, touching regions linked to Jesus' life and beyond? Scientists are cautious, emphasizing that despite the rich genetic evidence, definitive conclusions remain elusive without corroborating archaeological and historical context. This unexpected biological message inside the shroud invites us to reconsider its provenance and significance. It portrays the cloth as a vessel carrying a hidden history embedded within its threads, a story written in the language of DNA, invisible to all until now. The shroud's fabric itself appears to be a living record, silently witnessing the passage of people across centuries and continents. The image that emerged in 1898 would become a focal point, capturing imaginations and challenging skeptics in unexpected ways. Historical context and early discoveries. The modern fascination with the Shroud of Turin began in a most unlikely way, with a single photograph taken on May 28, 1898. A lawyer and amateur photographer named Secondo Pia was granted rare access to capture images of the shroud during a public exhibition in Turin, Italy. Photography at that time was far from simple. Bulky equipment, long exposures, and complicated chemical development marked the process. Yet, when Pia developed his photographic plates in the solitude of his dark room, something extraordinary appeared. Instead of the faint, ghostly outlines one might expect, the photographic negative revealed a clear, detailed human face. Features like deeply set, closed eyes, a broken nose, a moustache, and a forked beard emerged with startling realism and emotional gravity. Marks of bruising and suffering were evident on the cheeks, transforming the image from a mere cloth impression into something far more vivid and lifelike. This moment reshaped the way the world viewed the shroud, suggesting it captured not just an outline, but the visage of a real human being. What puzzled Pia and everyone who saw his photographs was the nature of the image itself. Unlike typical paintings, the shroud's cloth behaved like a photographic negative. The tones reversed on film produced a natural-looking portrait. But the technology to create photographic negatives would not exist for another 8 centuries. How could such a perfect negative be inscribed on linen long before cameras, film, or photographic chemistry? The shroud defied all attempts at classification because it functioned as both a medieval artwork and a forgery. The artwork presented itself as a time snapshot which no 13th century artist could replicate. The image served as an introduction to the cloth because the fabric would proceed to disclose additional hidden secrets which existed throughout its entire structure. The shroud existed through its entire history until Pia took his photograph because its past contained both empty spaces and mythical tales. People considered it a sacred religious artifact because they thought it contained Jesus Christ's burial cloth, yet its documented history remained incomplete and contested. The public first became aware of the object during the Middle Ages, when it became a significant presence throughout European territories. Yet existing tales and Byzantine records suggested that it had existed for a longer period. The shroud's complex past created additional secretive elements, which obscured its actual ownership history. Pia's discovery led to a scientific investigation which continued into modern times because it created base questions that scientists would solve with molecular biology and genetics research. Where had the cloth been? The cloth contained which faces and traces? What actual significance did the image hold? From these initial revelations flowed further investigations into another subtle and telling category of clues: microscopic botanical evidence embedded in the fabric, botanical clues, and geographic origins. Beyond DNA, the Shroud of Turin carries more silent witnesses of history, microscopic traces of plant life trapped within its threads. Scientists studying these botanical clues identified 58 distinct plant species on the cloth, marking it as a biological archive of the many regions the shroud encountered. While 17 species were native to Europe, unsurprisingly reflecting centuries of handling there, the rest came from much farther afield, spanning the Middle East, Turkey, and Anatolia, tracing a path that aligns closely with the shroud's suspected historical journey. Among these plants, researchers focused on pollen grains, especially those growing only in very specific regions between Jerusalem and Jericho. One particular species, Gundelia tournefortii, a thorny desert plant, was found in notably high concentrations near the head and shoulder areas of the shroud. This plant's sharp, long thorns are remarkably similar to those described in biblical accounts of the crown of thorns placed on Jesus during his crucifixion. Its bloom season in spring, near Passover, further reinforces the symbolic and historical resonance. Another key species identified was Zygophyllum dumosum, a plant native almost exclusively to the Judean desert and parts of the Sinai Peninsula. These botanical markers are difficult to dismiss as accidental contamination or medieval forgeries, especially as obtaining such pollen would have been virtually impossible for a European artist in the 13th century. Collecting and dispersing microscopic grains from such geographically restricted plants would require proximity to those very landscapes. These floral fingerprints act much like a forensic breadcrumb trail, pointing the relic back along ancient trade routes and pilgrimage paths historically connecting regions central to early Christianity. Cities like Edessa, along the Silk Road, and Constantinople come to mind. major crossroads where the shroud was reportedly housed and venerated for centuries. The diversity of plant life complements the genetic evidence, reinforcing the idea that the shroud traveled widely and absorbed environmental traces from numerous regions. It suggests the cloth was not confined to one place or era, but was carried through complex networks of human movement, earning its layered biological story. This botanical evidence also complicates simple medieval origin theories. Combined with other scientific data, it paints a picture of a cloth deeply rooted in the geography and history of the ancient Near East, rather than created in a European workshop. The growing body of scientific evidence from DNA and plants leads naturally to the examination of what those ancient bloodstains might reveal about the person wrapped in this enigmatic linen. The bloodstains and biochemical evidence. The Shroud of Turin holds more than just faint impressions and pollen grains. It carries actual traces of human blood, and modern science has unraveled astonishing details about its nature. In 2017, a research team, led by Giulio Fanti at the University of Padua, applied advanced techniques like transmission electron microscopy and Raman spectroscopy to analyze the stains. Far from pigment or paint, the microscopic analysis revealed genuine human blood embedded within the fibers. This blood was identified as type A, a relatively rare blood group, notably matching findings from another relic known as the Sudarium of Oviedo. What makes this blood extraordinary are its chemical markers. Scientists found unusually high levels of creatinine and ferritin bound to hemoglobin, substances often elevated after extreme physical trauma and severe stress. These signatures suggest the person wrapped in the shroud endured intense injuries before death, likely consistent with scourging and crucifixion practices known from Roman times. The blood chemistry here goes beyond visual resemblance. It records biological evidence of suffering at a molecular level, something impossible to fake with medieval techniques. Another striking aspect is the blood's color retention. Unlike ancient blood that typically darkens to brown or black over time, the stains on the shroud remain reddish. Researchers believe this is due to high levels of bilirubin, a compound produced during extreme trauma that can preserve the red hue longer than usual. This detail aligns closely with theories that the cloth enveloped an individual who underwent profound physical agony shortly before death. These biochemical clues reinforce the view that the shroud is far more than an artistic creation. They indicate an intimate connection to a severely wounded human body, one wrapped in linen with wounds that left genuine physiological traces. A painter might replicate the appearance of blood or bruising, but creating the complex molecular signature identified in these stains would have been beyond any medieval or even modern forgery. The fabric contains bloodstains which match the distribution patterns found in historical accounts of crucifixion. Marks appear in locations consistent with nails through the wrists instead of palms, and lacerations correspond to those inflicted by Roman scourges. The anatomical details of the shroud demonstrate that it contains actual crucifixion wounds, which exist as real evidence from the crucifixion event. The blood evidence, when combined with genetic and botanical data, provides evidence about a specific geographic area and historical time period. The biochemical signatures establish a link to the ancient Near East during the early Christian period. The researchers believe that the shroud displays physical realities from a time and location which parallels the crucifixion narrative. The evidence fails to resolve all inquiries, yet it creates more uncertainty about how the shroud originated and maintained its biological evidence of trauma. The evidence requires skeptics to analyze the difficult nature of the stains, which cannot be explained through standard art history and forgery methods. The profound nature of the blood evidence naturally draws attention to investigations into the shroud's age, where complex debates over carbon dating continue to reshape our understanding. Carbon dating controversies and renewed debates. For decades, the dating of the Shroud of Turin has stood at the center of debate, crystallized by the carbon-14 tests conducted in 1988. Laboratories at Oxford, Zurich, and Arizona dated the cloth to between 1260 and 1390, firmly placing it in the medieval period. This result seemed, at first, a decisive blow to claims of its ancient origin and connection to Jesus Christ. The findings led many to view the shroud as a medieval religious icon, rather than a genuine burial cloth from the 1st century. However, the overwhelmingly accepted date soon faced serious scrutiny. Critics pointed out that the sample used for carbon testing was taken from the cloth's outer edge, a section heavily handled during centuries of exhibitions and possible repairs. The corner had absorbed sweat, oils, dust, and other contaminants, potentially skewing the results. Chemical analysis by Raymond Rogers from Los Alamos National Laboratory added weight to these concerns, suggesting that this edge might include threads from medieval restorations conducted by the Poor Clare nuns. Rogers's findings indicated that new material interwoven with the original linen could explain the younger carbon dating. This raised the disturbing possibility that the 1988 samples did not represent the true age of the entire shroud, but rather a later patch. If so, the medieval dating would reflect repair work rather than the original fabric, leaving the question of the shroud's age wide open. Years later, fresh methods attempted to untangle this mystery. In 2022, Italian physicist Liberato De Caro applied wide-angle X-ray scattering, WAXS, to analyze the molecular structure of the linen. Instead of measuring contamination like conventional carbon dating, WAXS examines how the fabric's molecules change as they age. When compared with textiles from known periods, the shroud's structure seemed closer to 1st century linen than to medieval cloth. This approach reignited debate, challenging the assumption that the shroud was simply a medieval creation. The possibility that it might far predate the carbon dating results opened new avenues of scientific and historical investigation. If correct, it would align with both the DNA and botanical clues pointing toward ancient Near Eastern origins. Yet, even with these advances, no consensus has been reached. The dating problem remains tangled in layers of history, preservation challenges, and the complexities of textile analysis. Each new test answers some questions, but often raises others, illustrating how the shroud defies easy classification. This controversy over age highlights the broader challenge of interpreting a relic so steeped in human contact and care. It tests the limits of scientific methods and underscores how preservation and restoration can complicate efforts to pinpoint origin. The extraordinary scientific inquiries into the shroud's blood and age set the stage for confronting its most confounding feature, the image itself, which continues to defy explanation even today. The unsolved enigma of the image. One of the most baffling aspects of the Shroud of Turin is the mysterious image that decorates its surface, an image unlike any conventional artwork or photograph. Scientists have long searched the cloth for traces of paint, dye, or pigments that might explain the figure's delicate shading and detail, but found none. Instead, the image exists as a faint discoloration only on the outermost surface of the linen fibers, within a layer thinner than a human hair. This subtle chemical change, likely caused by oxidation and dehydration, remains unexplained by any known artistic process predating photography. Attempts to replicate the image have largely failed. Researchers at the Italian National Agency for New Technologies, Energy, and Sustainable Economic Development experimented with heat, chemicals, and different types of radiation. While some techniques, like brief ultraviolet light pulses, came close to producing similar effects, they required modern equipment far beyond what could have existed in ancient or medieval times. This gap between technology available in the past and the image's characteristics fuels questions about how it was created. In 1976, a NASA imaging team analyzed photographs of the shroud using the VP8 image analyzer, a device designed to render three-dimensional objects from two-dimensional images. Unlike ordinary pictures or paintings, the shroud's image surprisingly contains genuine three-dimensional information. The brightness variations relate closely to the distance between the cloth and the human body it supposedly covered, a feature unprecedented in traditional art and one that challenges conventional explanation. Further examination of the figure's wounds and markings adds more complexity. The placement of the nail wounds through the wrists instead of the palms aligns with Roman crucifixion practices, indicating a realistic understanding of ancient methods. Other subtle details, such as shapes resembling coins over the eyes from the time of Pontius Pilate, hint at historical accuracy that seems difficult to fabricate. These interwoven bits of evidence deepen the image's mystery beyond its mere appearance. Scientists from different disciplines, which include biology and chemistry and physics and archaeology and numismatics, continue their research on the shroud, which reveals new fascinating details through each study they conduct. The image cannot be classified as either a forgery or a natural occurrence because it exists between the two categories. The work combines elements of art and scientific research and historical study, which creates difficulties for people trying to identify its precise nature. Although scientists have developed better tools and acquired more scientific knowledge, they still cannot explain how the image was created. Some researchers believe that natural chemical reactions occur after intense events, which include radiation bursts and human body decomposition, while others think that unknown phenomena exist. The image functions as a permanent observer, which records a secret that cannot be completely understood. The shroud presents an unbreakable barrier because it combines features of photographic negatives with three-dimensional imaging and the cloth's biochemical properties. The requirement needs research about undiscovered facilities that create artistic work, which merges scientific and artistic practices through methods that have never been used before. The shroud's mystery exists because the image serves as its main element, which holds the puzzling problem that continues to astonish scientists and believers. The Shroud of Turin is more than a relic. It's a complex scientific puzzle combining ancient biology, chemistry, and history. From hidden DNA to enigmatic images, each layer deepens the mystery. If you found this journey into science and history fascinating, please like and subscribe for more explorations that challenge what we think we know. Thanks for watching.